Hello student. Now we are going to discuss regulation of respiration or control of breathing. First of all, we are going to classify the various mechanisms which are responsible for regulation of respiration, right? So, mechanisms. Number 1, I'm writing here. The main mechanism is neural regulation of respiration, right? The second major mechanism is the chemical regulation of respiration, right? And the third one I'm writing here is the non-chemical regulation of respiration. Non-chemical regulation of respiration. Now, under this non-chemical regulation, we are going to discuss the various reflexes which are controlling our breathing, like for example Herring Brewer reflexes as well as the J receptor reflexes, right? So these two reflexes we are going to discuss at the end of this chapter, right? Under the chemical regulation of respiration, what we are going to discuss? Obviously, chemical regulation means we are going to discuss various chemoreceptors which are controlling our breathing. Now, whenever we'll be discussing chemoreceptor, you know that chemoreceptors are located mainly in two locations. One is peripheral chemoreceptors, right? So, peripheral chemoreceptors, as well as there are certain area of the CNS where the chemoreceptor are also located, that is known as the central chemoreceptor. Central chemoreceptor. So where these peripheral chemoreceptors are located, we have discussed this part in cardiovascular system also, that location of the peripheral chemoreceptor are at the level of carotid bodies as well as it is also located at the level of aortic bodies, right? So carotid body and aortic bodies are the peripheral chemoreceptor. Now how these chemoreceptors are stimulated, what is the mechanism, how they are controlling breathing, that we are going to discuss a little later. But before that, look at this central chemoreceptor is located at the level of medulla. Where in the medulla? At the ventral surface of the medulla. So on the ventral surface of the medulla, right? So these central chemoreceptors are located, and how this central chemoreceptor is going to control our respiration, that we will be also discussing a little later. So before discussing this chemoreceptors or chemical regulation of respiration, first we are going to discuss the major mechanism of respiration, that is the neural control or neural regulation of respiration. This neural regulation of respiration, again it is divided into two types. One is voluntary control of respiration, voluntary control of respiration, and second one is the automatic control of respiration. Automatic control of respiration, right? So voluntary control means we can control our breathing or respiration according to our wishes, own wishes. Like for example, if I ask you, hold your breath for some time, could you do that? Answer is yes. Okay. And if I ask you, hold your heart, stop your heart for some time, will you be able to do that? Answer is no. Why not? Because cortical control, the cortex controlling the respiratory center, but cortex has no direct control on heart. That's why it is said that voluntary control of respiration is done by cerebral cortex, right? So the cerebral cortex has influences on various respiratory center as well as it has also influences on spinal cord, because of which we can control our breathing according to our own wishes, and that is known as voluntary control of respiration, purely done by cerebral cortex. So automatic control of respiration, okay, who is responsible for this automatic control of respiration? Two important areas of the central nervous system, number one pons, and second one is the medulla, right? So automatic control center are located at the level of pons and medulla. Now, the upper part of the pons contain one important center that is known as pneumotaxic center, right? And the lower part of the pons contain another important center that is known as apneustic center, right? So pons contain two center. Upper part of the pons, right? So I'm writing here pons. Upper part of the pons contain pneumotaxic center and the lower part of the pons contain apneustic center. Both of these center, which are located at the level of pons, they are responsible for controlling the rate and depth of breathing. But if I ask you, which center is mainly for rate controller, your answer will be rate is mainly controlled by pneumotaxic center and depth is mainly controlled by apneustic center. So both the center are responsible for rate and depth control of breathing, but rate controller mainly means pneumotaxic center, depth controller main center means apneustic center. Now forget about rate and depth. If I ask you where from the respiratory rhythm is originating, who is the pacemaker of respiration, who is starting the respiration, right? So the bigger question is that respiratory rhythm is generated from which center? Your answer will be obviously medulla. But if you look at the level of medulla, again you have to remember another two important area. One is DRG, that is the dorsal respiratory group of neuron, right? Dorsal respiratory group of neuron, and second one is the VRG, ventral respiratory group of neuron. Now you have to be careful here. If you look at Guyton textbook of medical physiology, Guyton is saying that DRG is the main center for respiration. But if you look at the other books and if you look at the recent literature, then the concept has been changed a little bit. Nowadays, the main concept is that main respiratory center for rhythm generation is located at the level of VRG. But which part of the VRG? I'll tell you. But before that, look at what is the functions of this DRG, right? So this dorsal respiratory group of neuron, right? So this is a collection of neurons at the level of medulla which are located close to nucleus tractus solitarius. You must have heard this nucleus tractus solitarius name. So this is the center. NTS is the center for various afferent from various viscera, right? So this DRG is located close to this nucleus tractus solitarius, and the functions of this DRG is similar to this NTS, that it is the sensory afferent for respiratory center. So what I am writing, this DRG is the sensory integration center for respiration, right? What is the meaning of that? You will understand a little bit. Broadly, the meaning is that from different periphery, like for example, from the level of lung, from the level of muscles, various signals are coming to the respiratory center. These are the peripheral signals. Who is integrating all these peripheral signals? That is nothing but your dorsal respiratory group of neuron. According to the recent concept, this is the main function of DRG. Okay, it is not responsible for rhythm generation. So where the respiratory rhythm is generated, you know, that this ventral respiratory group of neuron, if you look at, it has four subdivision. Fourth subdivision means it has four parts, right? So fourth part I am writing here, number 2, number 3 and number 4, right? So all these four parts has different functions, and among these four parts, one part is there which is responsible for pacemaking activity of respiration. So to understand this, first of all, you look at the diagram which is given in books, right? So this is the diagram I have taken from a book. You first look at that, you are finding out this area, that colicar fuse area and paravicalis complex. Close to this, the center which is located is the pneumotaxic center, so upper part of the pons. So here also the pneumotaxic center will be located, right? Then just below this pneumotaxic center, somewhere here, the amniotic center will be present. The amniotic center will be present. Now you can find out the nucleus tractus solitarius clearly here. So this is the nucleus tractus solitarius, this green color. Close to this nucleus tractus solitarius, there will be a collection of neuronal group, and this collection of neuronal group is known as the dorsal respiratory group of neurons. So this is my DRG. Now try to find out the ventral respiratory group of neuron. So you can easily find out that there is a large area, large group of neuron here. This large group of neuron, this is nothing but your ventral respiratory group of neuron. Getting my point? So this is the ventral respiratory group of neuron. Now you try to identify the different parts of this ventral respiratory group of neuron, right? So first part, as you can find out here, this is the Böttinger complex, right? So this is the Böttinger complex. Then just after this Böttinger complex, you are finding out the pre-Böttinger complex. Then you are also finding out the rostral VRG and the caudal VRG. So what are the different parts? You are finding out Böttinger complex, then pre-Böttinger complex of neuron, then rostral VRG, and then the caudal VRG. So you can easily finding out that this ventral respiratory group of neuron is consist of 4 different clear zone. One group of neuron is known as the Böttinger complex. Okay, you know that this Böttinger complex was first time discovered by Jack Feldman. So Professor Jack Feldman, he is the first scientist who discovered this Böttinger complex. So you must be thinking, Böttinger, is it a name of scientist? The answer is no. The scientist who discovered this Böttinger complex of neuron name is Jack Feldman. So when the Jack Feldman found out that this Böttinger complex is very important center for controlling respiration, okay, in the scientific meeting when he was presenting his findings that we have found out a center which is very important center for respiration. So he told in the scientific meeting that I want to make this center very famous so that everybody understand the importance of this center, and I want to give a name to this center. So there was a white wine bottle kept on the table. You know, this Böttinger is nothing but the white wine, a very famous white wine. And Dr. Feldman just hold the bottle from the table and he told the center will be known as this Böttinger center, right? So that way this Böttinger is not the scientist, it is nothing but a white wine, and according to this white wine the center is named Böttinger complex. Now later on the same group, that is the Feldman and colleague, found out that it is not the Böttinger, but caudal to the Böttinger there is another center which is particularly more important for respiratory control, and that's why this center is known as pre-Böttinger. Ideally this center should be known as post-Böttinger because it is located just behind the Böttinger complex of neuron. But because this center was discovered later and found out that this is more important than even Böttinger also, to give importance to this center, it is known as pre-Böttinger complex. So from my discussion, now it is clear to all of you that the main center for respiration or main center for regulation of respiration is the pre-Böttinger complex, and that's why pre-Böttinger is known as pacemaker of respiration, right? So now I will write down all these 4 parts of VRG, ventral respiratory group of neuron. So tell me what are the different parts. So Böttinger complex of neuron, Böttinger complex of neuron, then second one is the pre-Böttinger complex of neuron, pre-Böttinger, then third one is the rostral VRG, and fourth division is the caudal, okay, VRG, right? Obviously, you have to remember all these four names. Now, if I ask you, what is the role of this Botzinger complex of neuron? You know that this Botzinger complex of neuron, it contains mainly the expiratory group of neuron, and the main role is to control the expiration. So these neurons are active. These neurons are active mainly during expiration. Mainly during expiration, right? So this way you have to remember Botzinger complex of neuron is for expiration. Then this pre-Botzinger complex, I told you this is the most important neuron, the rhythm of respiration. So I am writing here very, very important, the rhythm of respiration. Rhythm of respiration is generated at the level of pre-Botzinger complex neuron, because of which this is known as pacemaker of respiration. Please note down. I have seen 3-4 MCQ already has been asked from this same topic, that where from the respiratory rhythm is generated, where the pacemaker of respiration is located. So pacemaker means that this center can produce spontaneous inspiratory and expiratory rhythm, right? That's why this is known as pacemaker. Now this pacemaker neuron, if you look at the discharge pattern, okay, then you will find out that the discharge pattern occurs in a step-by-step fashion. Gradual increase in discharge of this neuron in a ramp fashion. That's why this is also known as respiratory ramp generation, RAMP, ramp signal generation, right? So if the question is respiratory ramp signal is generated from which area, again your answer is pre-Botzinger complex. What is the meaning of this? Meaning of this, that respiratory rate and depth, they never rise to a very sudden level, okay, sudden high level. There is a smooth rise of inspiration and smooth rise of expiration. So that is known as the step-by-step increment of discharge pattern of neuron, and that is ramp signal. This is also known as, this pacemaker is also known as, that these are the neurons which are responsible for central pattern generator, CPG neuron for respiration. CPG stands for central pattern, PATTERN, pattern generator neuron. They are also located at the level of this pre-Botzinger complex. So central pattern generator means inspiration followed by expiration, inspiration followed by expiration. This is a pattern, and this pattern is generated from the center, the CNS center. Which center? That is nothing but pre-Botzinger complex. So pre-Botzinger complex, respiratory rhythm generation, because of which it is pacemaker. The pacemaking activity generate nothing but the ramp signal. The pacemaking activity is also known as CPG, central pattern generator, right? Now you look at this Rostal-Weiergy. So this Rostal-Weiergy, it contains mainly the I group of neuron, that is the inspiratory neuron, and this neuron discharge during inspiration to causes dilatation of the pharynx and larynx. So what I am writing, this Rostal-Weiergy, they are responsible for dilation of pharynx and larynx during inspiration, right? So during inspiration, you know that there will be dilation of the pharynx and larynx, and that is dominantly done by this Rostal-Weiergy. So specific question can be given in future from this different area. And this Caudal-Weiergy, you know, this Caudal-Weiergy, they mainly contain the expiratory group of neuron, and this neuron dominantly discharge during forceful expiration. So whenever you are doing forceful expiration, you take help of the accessory muscles of respiration, right? You take help of accessory muscles of respiration. This control of the accessory muscles of respiration, like for example your abdominal muscles, like for example your internal intercostal muscles, all these muscles, they should be contracted during forceful expiration, and this control is done by this caudal part of the Weiergy. That's why I am saying that it is responsible for forceful respiration or forceful expiration. Okay, I have seen this question in JIPMAR MCQ, right? They asked that during exercise, the forceful respiration is controlled by which part of your center. Obviously, the answer is ventral respiratory group, but if they ask you which specific part, your answer will be caudal part of the ventral respiratory group, right? So this way, you have to remember all these neuronal center which is controlling your breathing. So now you look at this chart, okay, and you will find out that many MCQ can be simply framed from this chart only, that what are the different center which is controlling your respiration. Now, after understanding the role of this different center, we have to read about one clinical condition or interesting clinical condition that is known as Ondine's curse, right? You must have heard this Ondine curse, very, very interesting, right? So, this Ondines, this is a German mythology. According to this German mythology, Ondines was a very beautiful water nymph, but by mistake or anyway, she fell in love for a man which is nothing named as Hans, right? So Ondines fell in love for Hans, and Hans promises that he is not going to marry a second woman in its lifetime. But Hans was not faithful. So one day, Ondines came to know that Hans married a second woman. Because of this, Ondines get severely angered and she gave a curse to Hans that in your whole life you will not be able to sleep. What is the meaning of that? The meaning, Ondines wants to say that Hans will have no automatic control of respiration, no automatic control of respiration, but voluntary control of respiration will remain intact. Voluntary control of respiration will remain intact, right? So what is the meaning of this? Obviously, Ondines is not going to say this kind of medical terminology. So she simply said that you will not be able to sleep peacefully in your whole life. Now you look at, if there is no automatic control of respiration, means pontine and medullary center are gone, but which center are still intact? The cortical control is still intact. So cortical control means when you are awake, your cortex is awake and you can control your breathing, inspiration, expiration. But whenever you lie down and you go for sleep, so your cortical control will be lost because cortex will also go for sleep. And what will happen? Your respiration will stop. Whenever your respiration stop, the carbon dioxide will be accumulated in your body, that will produce dyspnea. So again you will wake up and again you will feel sleepy. Whenever you sleep, again you wake up. So what is happening? Hans is not able to take a peaceful sleep. Not only peaceful, sleeping and again waking up, sleeping and again waking up. This will continue until and unless there will be sheer exhaustion of all the respiratory muscle, and ultimately death will occur. So severe curse, right? All that is looking very simple curse, but the curse is very, very severe, and that is known as Ondine's curse. So what is the clinical version of this? That there are certain congenital disorder which is known as congenital central hypoventilation syndrome, and that is nothing but equivalent of this Ondine's curse. What I told, congenital central hypoventilation syndrome. But not only congenital, there are certain clinical conditions which can arise in case of adult life also. Like for example, if there is a bulbar poliomyelitis. So in case of bulbar poliomyelitis, not the spinal poliomyelitis, if the polio is affecting the medulla and pontine region, and if these centers are destroyed, then automatic control is lost and voluntary control is intact, and that will produce similar condition like that of the Ondine curse. Not only that, certain tumor at the level of medulla can also produce this kind of clinical picture, or even during after injury, this kind of scenario can also appear. So in that cases, the only support can be given is the ventilated support you have to give to the patient, right? So this way we have to remember the importance of this Ondine's cuts, right? Okay, so interesting, right? So it should be faithful, otherwise this Ondine cuts like scenario can happen, right? So now our main understanding that we are going to do now is the how this neuronal center, how they are connected with one another. So we have seen very important center, pre-Bourdinger complex, we have seen pneumotaxic, amniotic center. So how these centers are connected with one another, right? So to understand this thing, first of all, I am drawing one pre-Bourdinger complex neuron, right? So this box is nothing but the pre-Bourdinger complex, and just above this I am drawing another box. So this box is nothing but the cortex. So suppose this is your cerebral cortex and this is the pre-Bourdinger complex of neuron, right? Okay. So pre-Bourdinger, what is that? It is nothing but a respiratory pacemaker. So what do you mean by respiratory pacemaker? Means it can produce spontaneous inspiration, expiration. So what should be there inside the pre-Bourdinger complex? Neurons, obviously neurons, but what kind of neurons? There will be certain group of neuron which are known as I neuron, and there are certain group of neuron which are known as E neuron. So I for inspiration and E for expiration. Now remember, this neuron, they are spontaneously discharging neuron because of their membrane properties, because of certain channels on their membrane. But what is the particular membrane property by which it is producing spontaneous inspiration, spontaneous expiration? Till date, we do not know that. Like for example, if you look back in the cardiovascular system, we know the mechanism of SA nodal pacemaking activity, why that is generating spontaneous rhythm. But for this inspiratory and expiratory group of neuron, they are spontaneously discharging neuron. That's why they are known as the pacemaker neuron. This pacemaker property is due to certain membrane channels, but we do not know what are those channels, right? So remember, this I neuron and E neuron, they are pacemaking neuron. First, the I neuron will start discharging and it will remain active for 2 to 3 seconds. When the I neuron is discharging, it will have an inhibitory signal on E neuron, right? So what I am saying, so when the I neuron is discharging, it is inhibiting the E neuron. And how long this I neuron will continue to discharge? For 2 to 3 seconds. After 2 to 3 seconds, this E neuron will be activated automatically, and when E neuron is activated, it will suppress the I neuron. So there is a mutual inhibition between these two neurons, okay, and there is a pattern: inspiration followed by expiration, inspiration followed by expiration. How this thing is happening, what is the reason behind this, what is the molecular reason, we do not know, but this kind of pattern is there at the level of pre-Bourdinger complex, right? Now if you look at, then you will find out that this I neuron, they are extending up to the level of the spinal cord. Means the axons of this neuron, they are projected at the level of the spinal cord, and at the level of spinal cord they are influencing spinal neurons, right? So what is happening, I am drawing another box here. So suppose this is my spinal cord, right? So I told you the I neurons are extending at the level of the spinal cord. Which region of the spinal cord you are talking, sir? I am talking of the cervical region and the thoracic region of the spinal cord. So at the level of spinal cord, what this I neuron is doing? This I neuron is giving influences to the cervical region neuron. Which neuron? That is the phrenic nerve, right? And at the level of thoracic region, it is controlling the intercostal nerves and it is controlling the intercostal muscles. So intercostal nerves. Because of which you know that whenever this eye neuron is activated, look at, so when the eye neuron is activated, what will happen? This activation will go to the level of the spinal cord, and at the level of spinal cord it is going to activate this phrenic nerve, right, and the intercostal nerves. Because of which all of you can easily understand that your diaphragm, okay, is going to be contracted, as well as your intercostal muscles. Your intercostal muscles are also going to be contracted, and we all know that whenever your diaphragm and intercostal muscles are contracted, then what will happen? There will be expansion of the lung and chest wall. So lungs and chest wall will ultimately expanded, and that is nothing but our inspiration, right? So now we can understand that whenever this pacemaker neuron is discharging at the level of medulla, it is sending signal, active signal to the level of phrenic nerve and intercostal nerve, because of which your diaphragm and external intercostal muscles are contracting, and then your lungs and chest wall is expanding, and that is nothing but inspiration. How long this neuron will continue to discharge? 2 to 3 seconds. When this eye neuron is discharging, it is inhibiting which neuron? It is inhibiting E neuron. After 2 to 3 seconds, whenever this E neuron is activated, now it is going to simply inhibit the eye neuron, and whenever you inhibit the eye neuron, automatically your diaphragm will be relaxed and your intercostal muscles will also be relaxed, and that is nothing but expiration. That's why we say that expiration is a passive one, because during expiration you are not getting any direct influence on the spinal cord, but it is simply inhibition of the eye neuron which is sufficient to causes expiration to start. That's why we say that you are not getting any active signal in the phrenic nerve during expiration, so it is a passive process, right? So this way the pre-Bourdinger complex neuron is working. This way the pre-Bourdinger complex neuron are working. Now this pre-Bourdinger complex neuron, they are under the control of cerebral cortex, that is for voluntary respiration, so forget about cerebral cortex. The other two centers which are responsible for controlling this pre-Bourdinger is nothing but pneumotaxic and amniotic. So suppose this is the pneumotaxic center in the upper part of the pons, and this is the amniotic center in the lower part of the pons, right? Now this amniotic center, you have to remember one property of this amniotic center. A center stands for amniotic center. Amniotic center is a tonically active center. Tonically active center. What do you mean by that? Tonically active center means the center is always active until and unless you inhibit this center by some others neuron, right? So this way these A neurons, they are discharging always, continuously discharging. So who is going to inhibit this A neuron? There are two inhibitors, there are two brake to control this tonically active center. So who are they? First one, this pneumotaxic center has inhibitory control on this amniotic center, and there is another inhibitory control on this amniotic center. And what is that? That is through vagus nerve, where from the vagus is coming. Look at, so whenever your lungs and chest wall are expanding, you know that there are certain stretch receptor at the level of lungs, okay, at the level of smooth muscles of the lungs. So whenever these lungs are expanding, right, what will happen? It will activate the stretch receptor of the lung. It will activate the stretch receptor of the lung. This stretch receptor, they will put their signal at the level of dorsal respiratory group of neuron. I told you already that what is the function of this dorsal respiratory group of neuron? They are the sensory integration center. So now you look at from the periphery, the signal is coming. Where from the signal is coming? From the stretch receptor of the lung, where the stretch receptor are located at the level of smooth muscles of the bronchi and bronchioles. So whenever your lungs are expanding, it is activating the stretch receptor. Stretch receptor is sending signal to the DRG. So all these signals are coming through which neuron? It is coming through the vagus neuron. It is coming through the vagus neuron, and ultimately from this DRG the input is going at the level of amniotic center. And what kind of input? Inhibitory input. Getting my point? So please note down that this amniotic center is a tonically active center. So tonically active center means I require certain inhibitor who will keep in check the activity of this amniotic neuron. And what are these two breakpoints for this amniotic center? One is pneumotaxic center itself, and second one is the vagus nerve from periphery, which are coming from stretch receptor of the lung. So when this stretch receptor will be activated, obviously if there is stretch at the level of lung, how much stretch you are talking about? If your lungs are expanding, but how much expanding? Remember, whenever your tidal volume is going greater than 1 liter, right? So what is the normal tidal volume? 500 ml. If it is double or more than double, then and then only the stretch receptor of the lungs will be activated. Whenever this stretch receptor of the lungs is activated, it will put signal through the DRG, and from this DRG the inhibitory signal will inhibit the amniotic center of respiration. So although we are interested in amniotic center, so what is the role of this amniotic center? What the amniotic center is doing? Amniotic center has strong stimulatory control on this eye neuron. Now very important. Amniotic center, it is continuously stimulating the eye group of neuron more and more. So if it is stimulating eye group of neuron more and more, what will happen? Your inspiratory depth will be prolonged. Inspiratory depth will be prolonged. I told you, amniotic center is mainly for depth controller. So if you allow the amniotic center to control the eye neuron, it will continuously stimulate the eye neuron, and inspiratory depth will be prolonged. But that does not happen normally, because it is under the control of, it is under the check of two other center, that is the pneumotaxic center and stretch receptor of the lung. I hope you get it right. Now, apart from the stretch receptor of the lung, you know that we have discussed that chemical regulation of respiration is done by chemoreceptors. Okay, so chemoreceptors, particularly the peripheral chemoreceptor, okay, they are also giving their signal at the level of this DRG, and from the DRG the signal will go to the level of amniotic center as well as the pre-Borzinger complex. So what is the bottom line? Bottom line is that if you stimulate this peripheral chemoreceptor, what will happen? The stimulation will go to the level of this eye neuron or pacemaker neuron, but signal is going through which integratory center? That is nothing but your DRG. That's why I told you that DRG is the sensory integration center. Now, you know the role of amniotic center. Now you look at the pneumotaxic center. So what is the role of pneumotaxic center? If I stimulate this pneumotaxic center more and more, what is going to happen? Pneumotaxic center, obviously it will inhibit the A neuron. Inhibition of the A neuron means your eye neuron ultimately inhibited. A is connected with eye. So if you inhibit A neuron, ultimately your eye neuron is going to be inhibited. So inspiration will be inhibited. Not only that, pneumotaxic center has direct stimulatory influence on this E neuron. So please remember, not only inhibition of inspiration, but also stimulation of expiration is also done by pneumotaxic neuron. So whenever you stimulate the pneumotaxic neuron, your expiration will start. Expiration will be prolonged, and if I stimulate the A neuron, amniotic neuron, my inspiratory depth will be more and more prolonged, right? So this way you have to remember the functions of all this center. Now this connection is very important. First of all, you look at all this connection, right? Now next you have to understand certain MCQ which are based on experiment done on various animals, right? So how the scientist came to know that this is the function of amnestic center, this is the function of pneumotaxic center? Because of various experiment. So how those experiments are done, and in your MCQ they can directly ask you the experiment also. So questions will be like this, that if you give a cut above this Pons level, so if I give a cut above Pons level in an experimental animal, right? So there are some instrument by which you can cut at the different level of neuroaxis. So whenever you are giving a cut at upper part of the Pons, then I can understand that cortex is separated, means cortical control on other respiratory center is lost. So what is going to happen? What will be the result, right? So here I am writing cut above Pons. Okay, so whenever you give cut above Pons, your cortical control is lost. What is the role of cortex? Cortex is responsible for voluntary regulation, voluntary control of respiration. So loss of voluntary control of respiration. So loss of voluntary control of respiration, right? So this you have to remember, that if you give a cut at the upper part of the Pons, then loss of voluntary control of respiration, right? Then you give a cut at the mid part of the Pons. So now I am giving a cut here. So cut at the mid part of the Pons, middle part of the Pons, very important. AIMS has asked this question already. So cut at mid Pons, right? So questions will be given like this. So whenever you give a cut at the mid Pons level, you can understand the cortex is already lost, the pneumotaxic center is also gone. So control of the pneumotaxic center on this amnestic center is lost. So what was the control of this pneumotaxic center on amnestic? It was very important. It was like a brake. Pneumotaxic was inhibiting the amnestic center. Now that inhibition is gone. So what will happen to the respiration? I told you already repeatedly that respiratory depth will be prolonged. So I am writing here cut at the level of mid Pons. Okay, what will be the effect if you use a spirometer? If you use a spirometer and if you record this kind of breathing, so spirometer recording we have already discussed. Suppose this is the normal breathing of the animal, experimental animal. So this is the normal breathing. So whenever you give a cut at the level of mid Pons, just now you tell me the answer that there will be increase in depth of respiration. There will be increase in depth of respiration. So this kind of breathing will be there. But question is, the inspiration is more and more depth. Inspiratory depth is rising. But who is inhibiting the inspiration at this point? Who is inhibiting the inspiration at the top of this? Because of the inhibition of the inspiration, then and then only your expiration is starting. So my question is, who is starting the expiration? Who is inhibiting the amnestic center? So this increased depth is due to increased activity of amnestic center. So amnestic center, who is inhibiting it so that it can again allowing expiration to start. So look at, I told you that amnestic center has two negative brake. One brake is in the form of pneumotaxic center, which is already lost because you have given a cut, but this vagus neuron is still inhibiting the amnestic center. But how this vagus neuron will be activated? The vagus neuron will be activated when your stretch receptor are active, and when the stretch receptor are active, when your tidal volume is greater than 1 liter. Getting my point? So now we look at, whenever you give a cut at the level of mid Pons, your inspiration will be prolonged. So this is normal. Suppose this is 500 ml. It will be increased. Whenever your tidal volume is getting more than 1000 ml, at this point, the vagus nerve will be activated by the stretch receptor of the lung, and that will inhibit the amnestic center. Whenever you inhibit the amnestic center, your expiration will start like this. So this way, I can understand that if you give a cut at the mid part of the Pons, what kind of breathing you are finding out, I'll get a breathing like this where slow and deep breathing will be seen. So I'm writing here slow and deep breathing will be seen because of the loss of activity of the pneumotaxic center, right? So who is preventing this more deeper breathing? Vagus nerve. Now if I give a cut at the mid pons along with vagus cut, now you are cutting the vagus also, vagus cut also, right? Now you are cutting the vagus also. So now look at this experimental condition. So already you have given a cut at the level of mid pons, right? So this cut, and now you are giving another cut, that is to cut this vagus, right? So to cut this vagus, so whenever you cut this vagus, what is going to happen? Okay, now both your vagus is gone, your pneumotaxic center is gone, means both the break of the amnestic center is lost. So what will happen? Amnestic center, because it's a tonically active center, it will stimulate the eye neuron more and more. What will happen? Inspiration will be prolonged, and no one is there to start the expiration. So what will happen? There will be stop of respiration, there will be spasm of respiration at the maximum inspiration. So there is a spasm in between, there will be a little gasping effort to start the expiration, but expiration will not start until and unless death of the animal, right? So this way, whenever you cut the vagus also along with the mid pons cut, what is going to happen? There will be more and more prolongation of inspiration, and then there will be an inspiratory spasm at the apex of, at the top of maximum inspiration. This kind of phenomena is known as apneusis phenomena. So whenever your vagus is also cut, one kind of phenomena you are going to see, that is apneusis. What is apneusis? This is nothing but spasm of respiration, okay, at maximum inspiratory phase, right? So this kind of spasm of respiration at the maximum inspiratory phase is known as apneusis effect, right? So now you can answer here that if you give a cut at the level of mid pons, there can be two scenarios. I hope this is absolutely clear to you. Without vagus cut, so without cutting the vagus nerve, and with vagus nerve cutting, with vagus cut, right? Without vagus cut and with vagus, without, right? So if you cut vagus, just now we have seen what is the phenomena, apneusis is the phenomena, and if your vagus is intact, then slow and deep breathing is going to be seen. So this is very clear, I hope, and you have to remember this, you have to remember this information in your brain, right? That is the effect of cut at the level of mid pons, right? Now you look at the next cut, right? So now I am giving a cut here at the upper part of medulla like this. So now this is the cut where I am writing, this cut is going through the upper medulla, cut above medulla, right? Cut above medulla. So if you give a cut above medulla, what are the center that is separated from your respiratory center, right? So now I can see that my pneumotaxic center is gone, apneustic center is also gone. So pneumotaxic, apneustic, both the center is not connected with the pre-Bötzinger complex. So pre-Bötzinger complex is still intact. Okay, so can it maintain your respiration? Answer is why not? This is a pacemaker. So what do you mean by pacemaker? Pacemaker means it can continue your respiration, inspiration, expiration, inspiration, expiration. This can continue. So question is, so what is the role of this pneumotaxic and apneustic center then? I told you that pneumotaxic and apneustic, they are rate and depth controller. But without these two center also, respiratory rhythm can be maintained by pre-Bötzinger complex. So whenever you give a cut above medulla, if this is the normal respiration, so again I am drawing a spirometer recording. Suppose this is the normal respiration, Okay, whenever you give a cut above medulla, what is going to happen? Your respiration will look like this. Your respiration will look like this. So what you are finding out from this is your depth of respiration is constant. Is your rate of respiration is constant? Answer is no. Neither the rate, neither the depth is constant. So what kind of respiration this is? This is nothing but irregular type of respiration. Irregularly irregular. Rate and depth control is done by pneumotaxic and apneistic. Both of them are gone. So what will happen? Your rate and depth will not be fixed. So that's why you are not finding out that this tidal volume is not fixed. Even your rate is also not fixed. So this is nothing but irregular respiration. Now suppose I have got this effect when my vagus nerve is intact. Suppose this is the effect, irregular respiration. Okay, if I give a cut above medulla, I am getting an irregular respiration, right? But I am assuming that the vagus are intact. Now if I give a cut additionally at the level of vagus, what will be the additional effect? Will there be any additional effect? Look at where the vagus is giving its input. I told you, if you stimulate vagus, the vagus is going through this DRG and ultimately the vagus is giving its effect at the level of apneustic center. So if you give a cut here at the above the medulla, already the apneustic center is lost. So cutting the vagus will not have any additional effect if your cut is going above the medulla, right? So that way you remember that even if your vagus is also cut, suppose vagus is also cut, again the same thing, that you are going to get irregular type of respiration. So irregular respiration means this kind of effect will be seen, right? So that's why you know that if you give a cut above medulla, irregularly irregular respiration. Okay, vagus cutting has no additional effect. I hope you got it right. Now the next one or the last one is if you give a cut below this medulla. So spinomedullary junction, suppose there is an injury at the level of spinomedullary junction. What will happen? Your respiratory pacemaker is disconnected from spinal cord. So you can easily find out that if your respiratory pacemaker is disconnected from spinal cord, nothing, there will be instantaneous death. So cut below medulla, cut below medulla. Okay, no dramatic effect is seen. Okay, only there will be death, and that's why this is not a very famous MCQ, because you know all of you will be able to answer that if you give a cut below medulla, there will be instantaneous death. But the above medulla, midpoint of the pons, they are the very famous MCQ for your exam, right? So this way, remember that how these different neural center are connected with one another, right? So now if I show you the diagram, okay, so this is the diagram which is taken from genome. So the first, the cut number A, right? So this is the cut above pons, right? So if you give a cut above pons, what is going to happen? This is the normal respiration, right? Because I told you only voluntary control will be lost, nothing else. But look at these two graphs are given parallelly. One graph is vagus nerve is intact and another graph is vagus nerve is cut, right? So if the vagus nerve is intact, then cut above pons means nothing is going to happen, the respiration will remain absolutely normal. But if you give a cut in the vagus, whenever you give a cut at the level of vagus, vagus was inhibiting the apneustic center. So if you cut bilateral vagus, always the depth of respiration is going to be increased. So that way, if you cut vagus, what is going to happen? Increase in depth of respiration. So your answer will be slow and deep breathing, right? Slow and deep breathing. Similarly, this cut number B, this is the mid pons cut, right? We have already seen the mid pons cut effect. So this is the slow and deep breathing, and if you give a cut vagus additionally, then what is going to happen? This is nothing but your apneusis phenomena, right? So severe inspiration and then spasm of inspiration followed by death, right? So this is typically apneustic phenomena. And if this cut number C is above medulla cut, this is the cut above medulla. So if you give a cut above medulla, I told you already that there will be irregular type of respiration. So this is also irregular type of respiration. Whether you cut vagus or not, no significant effect is seen, right? And if you give a cut here, that is below medulla, okay? Below medulla cut means there is nothing but death, right? So that's why this is nothing but death. So this is all about your neural regulation of respiration, how the different neural center are controlling your respiration, right? Now after discussing the neural regulation of respiration, we are going to discuss the chemical regulation of respiration. So I hope remember that chemical regulation of respiration means it is done by various chemoreceptors which are present in our body. And I told you that chemoreceptor means one main group is the peripheral chemoreceptor and central chemoreceptor. And peripheral chemoreceptor, they are located at the level of carotid body and aortic body. So even if you forget the things which has been taught at the level of cardiovascular system, look at this, where the chemoreceptors are located. So suppose this is the internal carotid artery, right? And this is the external carotid artery. And this is the common carotid artery, right? So common carotid artery dividing into two, and we know that internal carotid artery at the point of origin there is a little dilated area. This is known as the carotid sinus. Okay. At the bifurcation of this internal and external carotid artery, you know that there are certain tennis racket shaped tissues are there, small tissues. The shape of this tissue is like that of a tennis racket, right? This tissue is known as carotid bodies, right? So this is nothing but carotid body, right? And this carotid body is nothing but the chemoreceptor, right? Similarly, if you look at the level of this arch of aorta, on the surface of this arch of aorta, again 2 to 3 tennis racket shaped structures are there, and these structures are nothing but known as aortic bodies, right? So this carotid body and aortic bodies, they are the peripheral chemoreceptor. Peripheral chemoreceptor. All of you know that in the same area the baroreceptors are also located, which we have already discussed in cardiovascular system, that at the level of this carotid sinus wall your baroreceptors are located, and at the level of this aortic arch wall your baroreceptors are also located, right? So this is the same area where this baroreceptor and chemoreceptor are located, but chemoreceptors are present within this carotid body and within these aortic bodies. So question is, what is there inside this carotid bodies and aortic bodies? So if you look at the level of this carotid bodies and aortic bodies, although the tissue is very small, only 2 millimeter in diameter, okay, and 13 milligram is the weight and 2 millimeter in diameter, but you know that this small tissue contains two different types of cells, right? So one cell is there which is almost round in shape, and surrounding this there is another type of cell which is protecting this round cell. So this blue colored cell, this is known as the type 2 glomus cell. So this is nothing but type 2 glomus cell, GLOMUS, glomus cell, right? And this red colored cell, this is nothing but the type 1 glomus cell. So if you look at the carotid body or aortic body, this body is nothing but it's a small tissue within which two main types of cells are there, type 1 and type 2 glomus cell. Between this type 1 and type 2 glomus cell, you know, type 1 glomus cell is the main sensor cell of chemoreceptor. What do you mean by sensor cell? You know, chemoreceptor means they are activated by certain chemicals of the arterial blood. Look at. So whenever the composition of certain chemicals is changed at the level of this arch of aorta, or composition of certain chemical is changed at the level of this carotid arterial blood, then the cells which are located inside this carotid body and aortic body, that is the glomus cell type 1, they can senses this chemical. That's why this is known as a sensor cell, right? So actual chemoreceptor is nothing but because of this sensor cell. So if it is question is given like this, that chemoreceptor are due to which of the following cells, your answer will be sensor cell, that is type 1 glomus cell. So what kind of chemical they can senses, or what are the activator of this sensor cell? So number 1, decrease in partial pressure of oxygen in the arterial blood, increase in partial pressure of carbon dioxide in the arterial blood, or increase in H plus ion in the arterial blood. So that is nothing but acidosis, right? So I am saying here that hypoxia, right, hypercapnia and acidosis, these three are the dominant stimulant for peripheral chemoreceptor, both carotid body, aortic bodies. But carotid bodies chemoreceptor are more sensitive than this aortic body's chemoreceptor, right? So these are the three stimuli, okay, which are responsible for stimulating this type 1 glomus cell. So what is the role of this type 2 glomus cell? Type 2 glomus cell, they are surrounding, they are protecting this type 1 glomus cell. So that's why they are known as supporting cell. They are known as supporting cell or sustentacular cell. Sustentacular cell, right? So they are known as a supporting cell or sustentacular cell for type 1 glomus cell, right? So this type 1 glomus cell, whenever these chemical signals are present at the level of blood, like for example hypoxia, hypercapnia or acidosis, this type 1 glomus cell will be activated, and whenever this type 1 glomus cell is active, what will happen? It will activate certain neurons which are present at the level of this glomus cell. So there will be certain neuron here. So these are the neurons, suppose these are the neuron which are connected with this type 1 glomus cell. So question is, what is this neuron? Okay, so this neuron is nothing but 9th and 10th cranial nerve, because if you look at here the aortic region, so which cranial nerve is connected here? The vagus nerve. And if you look at this carotid region, then it is nothing but 9th cranial nerve or the glossopharyngeal nerve is connected. So these two neurons, they are the afferent from this chemoreceptor. So whenever these chemoreceptors are activated, it will stimulate the afferent neuron which are connected with this glomus cell, and this afferent neuron ultimately stimulate the ventilatory center or respiratory center, that is nothing but pre-Bodenzinger complex. Getting my point? So now we have to understand that what is the mechanism of activation of this glomus cell? How glomus cell is activated in presence of this kind of signal, right? So suppose this is the diagram of a glomus cell, right? Glomus cell, which type? Type 1, right? It will be activated by certain chemical signal in the blood, hypoxia. Let's start with hypoxia. So this glomus cell, you know that this cell contain a channel, one ion channel, right? And that is very, very important. You must have to remember, type 1 glomus cell contain one ion channel. That ion channel is known as oxygen sensitive, right? Potassium channel, oxygen sensitive potassium channel, right? This has been asked in MCQ already, so please be careful. So normally this channel is open, and whenever the channel is open, we all know that if the potassium channel is open, potassium always goes out of the cell because of the concentration gradient. So potassium is going out normally when the oxygen tension in the arterial blood is normal, right? Now suppose the arterial blood which is supplying at the level of this glomus cell, it is having low oxygen tension, hypoxia. So whenever there is a hypoxia at the level of this arterial blood which is supplying this glomus cell, what is going to happen? This hypoxia will directly inhibit the oxygen sensitive potassium channel. So this hypoxia will directly causes closure of this oxygen sensitive potassium channel. I am writing here inhibition. So whenever there is a closure of this oxygen sensitive potassium channel, potassium will stay back inside the cell. Potassium will not be able to go out. So whenever potassium is staying back inside the cell, potassium is a positively charged. I can understand that more and more positively charged will be collected inside the cell membrane. More and more positively charged collection inside the cell membrane means which kind of voltage will be collected? Positivity inside the cell. So that is nothing but depolarization. This is nothing but depolarization. So closure of the oxygen sensitive potassium channel is leading to depolarization of the glomus cell. Whenever there is a depolarization, that there is another channel on the same glomus cell type 1, and this channel is activated in presence of depolarization, in presence of voltage. So obviously this is a voltage gated channel, but the channel is dedicated for which ion? The channel is dedicated for calcium ion. So that's why this is known as voltage gated calcium channel. It will open up, right? So whenever hypoxia is there at the level of glomus cell, hypoxia is causing closure to the potassium channel, that is causing depolarization. Depolarization is causing opening of this voltage gated calcium channel, right? So these are basically connected with one another, and this is opening the channel. Whenever the calcium channel is opening up, simple thing is that calcium is going to enter. Now in presence of calcium, that this glomus cell, it's a secretory cell. It contains multiple vesicles which contain various neurotransmitters. So whenever this calcium is entering inside this cell, these vesicles which are present inside this glomus cell, the vesicle will move closer towards the cell membrane, and then after moving close to cell membrane, what will happen? This vesicular membrane as well as this plasma membrane of the cell, they will fuse with one another, and that will leads to rupture of this area. Whenever there is rupture, I can easily understand that the neurotransmitter which is present inside this vesicle, suppose these are the neurotransmitter which are present inside these vesicles of the glomus cell, this neurotransmitter is going to be released, right? So in presence of calcium entry, I can understand that there is nothing but neurotransmitter release is occurring, right? So calcium is causing release of neurotransmitter, and this neurotransmitter will ultimately causes activation of the neuron which are connected here, which I already told you that which neuron is connected, 9th and 10th. If it is carotid body, then 9th neuron, and if it is aortic body, 10th neuron. So this neurotransmitter is ultimately going to stimulate this neuron, and because of the neuronal stimulation, action potential will be generated, and this action potential will ultimately travel at the level of pre-Bötzinger complex. Whenever this pre-Bötzinger complex is stimulated, what will happen? There will be increase in your ventilation. Increase in your ventilation means what parameter is going to be increased? Both the rate and depth of respiration will be increased. So this way, whenever you stimulate the chemoreceptor, right? So now you can understand the mechanism that in presence of hypoxia, whenever the peripheral chemoreceptor is stimulated, what is happening ultimately? There is increase in ventilation, hyperventilation. There will be increase in rate and depth, right? So if there is an hypoxia in my body, I will do like this. So this is hyperventilation. Both my rate and depth is rising. Now you know the mechanism that how the hypoxia is stimulating your ventilation. So the main cell which is detecting this hypoxia is the glomus cell type 1, and how the glomus cell type 1 is activated by closing of one of the main channel, that is oxygen sensitive potassium channel. Now the last question. I told you neurotransmitter release. What is the main neurotransmitter released by glomus cell type 1. If you look at Genong is writing that dopamine, but that's not absolutely correct. Dopamine is also there, but according to the recent literature, particularly if you open up Guyton is clearly saying, and the recent literature are also saying, that adenosine triphosphate is the main neurotransmitter of type 1 glomus cell, followed by acetylcholine, followed by dopamine, right? So please remember that if the question says the main neurotransmitter from this glomus cell type 1, answer is adenosine triphosphate, followed by acetylcholine, followed by dopamine. So this way the peripheral chemoreceptor are activated. So this is about hypoxia. So now if I ask you, sir, now I can understand that hypoxia, how it is stimulating the glomus cell. How the hypercapnia and acidosis, they are also doing the same thing. So whenever you think of hypercapnia and acidosis, they also causes closure of the same channel. They also causes closure of the same channel, that is the oxygen sensitive potassium channel. But this is done by certain indirect mechanism. Indirect mechanism. What do you mean by indirect mechanism? Means this hypercapnia and acidosis, they will activate or inhibit certain molecules which are present inside the cell in the form of second messenger, and those second messenger is ultimately going to cause closure of the oxygen sensitive potassium channel. That's why I said indirect. But what about hypoxia? The channel is sensitive to oxygen. That's why hypoxia can directly cause closure of the oxygen sensitive potassium channel. So the effect of hypoxia is the direct effect, and effect of hypercapnia and acidosis is indirect effect. So that's why, although all these three stimuli are doing the same thing, but if your question is like this, that among these three stimulus, which one is the primary stimulus for peripheral chemoreceptor? Which one is the primary stimulus for peripheral chemoreceptor? So obviously your answer is hypoxia. Obviously your answer is hypoxia. But if I ask you that primary means, the question can also be asked like this, the direct stimulus. But if I ask you what is the indirect stimulus, then carbon dioxide and acidosis. But if I ask you which one is the most sensitive stimulus for peripheral chemoreceptor, then the question is different. Most sensitive means if I change 1% of this oxygen, 1% change of oxygen, 1% change of carbon dioxide, and 1% change of H plus ion, which one is going to stimulate more? That is the sensitivity, right? So it is clearly written in book that most sensitive stimulus for peripheral chemoreceptor is hypercapnia, right? So please remember, primary direct stimulus for peripheral chemoreceptor is hypoxia. Most sensitive stimulus for peripheral chemoreceptor is hypercapnia. But sometimes this kind of question is also given, that most potent stimulus for peripheral chemoreceptor, your answer will be cyanide poisoning, right? Why cyanide poisoning? Cyanide poisoning also causes one type of hypoxia, which has been discussed in chapter of hypoxia later on, right? So that's why the cyanide, by causing severe hypoxia, histotoxic type of hypoxia, they are the most potent stimulus for peripheral chemoreceptor. Got it? Okay. So this is all about your peripheral chemoreceptor. Now central chemoreceptor, right? So what I told, where the central chemoreceptors are located? So central chemoreceptors, right? They are located at the level of ventral surface of medulla. I told you. So please don't forget that they are located at the level of ventral surface of medulla. Okay, just beneath the surface of medulla, on the ventral surface, this central chemoreceptor are located. So we are trying to discuss this central chemoreceptor with the help of this diagram, right? Suppose this is the cross section of the medulla, right? Okay, and surrounded by cerebrospinal fluid. So this is the CSF region, right? At the level of ventral surface of medulla, just beneath the surface, there are certain neuronal collections are there. There are certain neuronal groups are there, which are known as the chemosensitive neurons. So look at, I have made one neuron here, but suppose that there will be a group of neurons. These neurons are chemosensitive neurons. And these chemosensitive neurons are nothing but central chemoreceptor. So it is not like glomus cell. Glomus cell is not there at the level of medulla, obviously. Okay, neurons will be there. Some neurons can detect certain chemical signals and they are chemosensitive neuron. That's why this region of the ventral surface of the medulla is known as chemosensitive zone. Okay, so this is known as chemosensitive zone. So basically, central chemoreceptor is nothing but, it is nothing but the chemosensitive zone which is located at the ventral surface of medulla. There are certain neurons. But these neurons, they cannot detect the hypoxia, right? So if I ask you, which chemical can activate this central chemoreceptor? Very important. Never forget this. Okay, very important thing is that this central chemoreceptor, the main stimulus for central chemoreceptor is H plus ion, but H plus ion of cerebrospinal fluid or interstitial fluid of brain. So what I am saying, that these neurons, which I told that these are the chemosensitive neuron, this neuron will be stimulated only and only by H plus ion, but that H plus ion not from the blood. That H plus ion has to be present either at the level of this CSF, or this has to be present surrounding the neuron. So surrounding the neuron, which kind of water, which kind of fluid is there? That is the interstitial fluid of brain. So that's why I told you that H plus ion, either it has to be present at the level of CSF or it has to be present at the level of interstitial fluid of brain. Otherwise, the H plus ion will not be able to stimulate this central chemoreceptor. Getting my point? So you remember this. Okay. Now question is, why not hypoxia is a stimulus for this neuron, right? So now you look at, so suppose this is a capillary at the level of CNS, right? Capillary at the level of CNS. Suppose there is a decreased oxygen concentration at the level of this capillary. So suppose there is hypoxia at the level of this brain capillary. So whenever there is an hypoxia at the level of brain capillary, I can understand that this hypoxia is also going to affect the neuron which are located at the level of chemosensitive zone. Okay, so hypoxia will be there surrounding this neuron also. But is there any oxygen sensitive channel on the surface of this neuron so that it can detect hypoxia? Answer is no. So can it detect hypoxia? Answer is no. That's why remember, hypoxia, although it is a stimulus for peripheral chemoreceptor, but hypoxia is never a stimulus for central chemoreceptor. So hypoxia stimulate which chemoreceptor only? Stimulate only and only the peripheral chemoreceptor. Stimulate only peripheral chemoreceptor. Okay. Rather than stimulation, hypoxia can causes depression of central chemoreceptor, right? It can causes depression on central chemoreceptor, but not the stimulation. Stimulation is only and only at the level of peripheral chemoreceptor. Central chemoreceptor, if the hypoxia is severe, then they can get depressed, but never it will be stimulated, right? So if the question is like this, that one person has ascended to an high altitude, so there will be hypoxia and the person will start hyperventilating. So the person will start like this. So I can understand that the chemoreceptor is stimulated. But if the question is high altitude, hyperventilation is due to which kind of chemoreceptor? Your direct answer is only and only peripheral chemoreceptor stimulation, not the central chemoreceptor, because central chemoreceptor cannot be activated by hypoxic stimuli, right? Okay. Now, second. So Next stimuli for peripheral chemoreceptor, we have seen that suppose there is increase in H plus ion in the plasma. So that is acidosis. H plus ion is high at the level of blood. So that is acidosis. Can acidosis stimulate central chemoreceptor? Central chemoreceptor can be activated by H plus ion. But that H plus ion should be present at the level of CSF or interstitial fluid of brain. Now my question is, can this H plus ion from the blood can reach up to the level of this chemoreceptor neuron? Answer is no. Why? Because this H plus ion, it's an ionic molecule. Can ionic molecule penetrate through the brain capillary? Answer is no, because the brain capillary are protected by a strong barrier, and that is nothing but our blood brain barrier, right? So because of the presence of this blood brain barrier, what will happen? The H plus ion, because it is an ionic molecule, it cannot penetrate the blood brain barrier, and it will never be able to affect the central chemoreceptor. So that's why this is very peculiar, and this is the only main concept that you have to remember, that in case of acidosis, although the H plus ion is high inside your blood, but that H plus ion will not be able to cross the blood brain barrier. That's why it will not be able to stimulate the central chemoreceptor. So acidosis stimulate only and only which chemoreceptor? The peripheral chemoreceptor, right? But acidosis, it cannot stimulate central chemoreceptor. Why? Because of the presence of blood brain barrier. Right? Now you look at the third stimulus for peripheral chemoreceptor. What was the third stimulus? The third stimulus for peripheral chemoreceptor was the hypercapnia. So suppose there is increased concentration of carbon dioxide in the arterial blood. Can it activate or stimulate central chemoreceptor? So let's see. So suppose carbon dioxide is very high at the level of blood. Can this carbon dioxide cross the blood-brain barrier? Answer is why not? It's a gaseous molecule. So this gaseous molecule carbon dioxide will easily penetrate this blood-brain barrier. It will reach in the brain interstitial fluid. Not only that, it can further penetrate at the level of CSF. And at the level of CSF, this carbon dioxide is going to react with the H2O, and in this reaction will produce carbonic acid, and this carbonic acid will be divided into H plus ion as well as bicarbonate ion. And now this H plus ion, they are going to stimulate this central chemoreceptor. That's why I told you that whenever carbon dioxide is high in the peripheral blood, it can generate high amount of H plus ion at the level of CSF also, even at the level of interstitial fluid of brain also. And because of this H plus ion generation, it can easily stimulate the chemosensitive zone or central chemoreceptor. So we have previously seen that H plus ion or acidosis, okay, is an indirect stimulus for peripheral chemoreceptor. Now we have seen that carbon dioxide, it is stimulating the peripheral chemoreceptor also, as well as it is also stimulating the central chemoreceptor through production of H plus ion at the level of CSFs. So I can write down here that carbon dioxide, it stimulate, okay, both the peripheral chemoreceptor also, as well as it can also stimulate the central chemoreceptor also, peripheral and central. But if you look at the total stimulation which is done by carbon dioxide, then 80% or the major stimulation of carbon dioxide goes through the central chemoreceptor, and only 20% stimulation goes through the peripheral chemoreceptor. So suppose one question is given like this, that one person is hyperventilating due to huge amount of carbon dioxide in the body, or due to hypercapnia, right? So due to accumulation of carbon dioxide, the person is hyperventilating. What is the main stimulus? Your answer is the central chemoreceptor is the stimulus, right? Okay, peripheral chemoreceptor is also stimulated, but 20% of the total. So this way I have to remember that among these three stimuli, carbon dioxide or hypercapnia is the only stimuli which can activate both the peripheral as well as central chemoreceptor. So carbon dioxide is very, very important, right? Okay, you have to remember this carbon dioxide effect. Okay, now one applied aspect about this chemoreceptor. So this is the mechanism of central and peripheral chemoreceptor we have discussed. Now you have to remember one important point, that in case of COPD patient, okay, this chemoreceptor sensitivity decreases, not increases, remember. So there will be decrease in sensitivity, of chemoreceptor. Okay, this is known as desensitization of chemoreceptor. So what will happen? Your chemoreceptor is less sensitive. So what will happen in case of emphysema patient or in case of COPD patient, because of this insensitivity, because of this decreased sensitivity, carbon dioxide will not be able to stimulate the peripheral chemoreceptor because the sensitivity of the receptor is decreased. So what will happen? Gradually the carbon dioxide will be accumulated in patients. That's why we have seen that there will be increase in carbon dioxide accumulation in case of COPD patient, chronic COPD patient, because the peripheral chemoreceptor is desensitized. Otherwise, if the carbon dioxide is high, peripheral chemoreceptor will be stimulated and patient will hyperventilate the carbon dioxide. But in case of COPD, because of the chronic nature, because of the chronic hypoxia, the receptor get desensitized, right? Second applied importance. During voluntary hyperventilation, what happens in the body? So if I continue to do voluntary hyperventilation, right, suppose I am doing like this for the last 15 seconds. So whenever you are doing voluntary hyperventilation, what you are doing, more fresh oxygen is entering in your body and you are washing out the carbon dioxide from the body. So there will be more and more wash out of carbon dioxide from the body. So whenever more and more carbon dioxide is washed out, what will happen? It will leads to alkalosis, and this alkalosis will causes stopping of your peripheral chemoreceptor. This alkalosis will be inhibiting your chemoreceptor, and whenever your chemoreceptor is inhibited, what will happen? At certain point of time, there will be a temporary stoppage of your respiration, and that is known as apnea. That's why if you continue to do voluntary hyperventilation for some times, there will be a phase when your respiration will be stopped for few seconds, and that is apnea. So why the apnea is occurring? That is due to washing out of carbon dioxide. Okay, so carbon dioxide is not there in the body, alkalosis is also occurring in the body, and both are inhibition of the peripheral chemoreceptor. So your respiration will stop for certain period, and whenever your respiration is stopped, whenever the apnea is occurring, what will happen? Now gradually the carbon dioxide level in your body will start rising, because carbon dioxide will start accumulating in your body, and again it will start your ventilation. So that way, after this apnea, again you will start your ventilation, right? But there is a question that during this voluntary hyperventilation, if the subject is inhaling 5% carbon dioxide, so the person is taking a inhalation. So during this voluntary hyperventilation, subject is taking inhalation of 5% carbon dioxide in the gas mixture. So person is taking atmospheric air or oxygen, along with that he have given 5% carbon dioxide. So whenever I am taking inspiration, I am inhaling extra carbon dioxide, and whenever I am doing hyperventilation, it was supposed to wash out the carbon dioxide. But each time you are taking inhalation, you are taking this amount of carbon dioxide in your body. So what will happen? This carbon dioxide wash out is not going to happen if you are inhaling carbon dioxide containing air, right? Extra carbon dioxide containing air. So in these cases, the apnea is not going to happen. Apnea not seen, right? So this has been asked in MCQ, that voluntary hyperventilation with 5% carbon dioxide, what is the effect? Apnea is not going to happen. So these are the applied aspect of this chemoreceptor, right? Now we are going to understand how the carbon dioxide and how the oxygen, that is hypoxia, is stimulating this chemoreceptor. What is the stimulating pattern, not the mechanism, right? So hypercapnia and peripheral chemoreceptor stimulation. What is the relationship, right? So to draw this relationship, to discuss the relationship, I am drawing one graphical curve here, okay, graph here. So the x-axis of this curve is carbon dioxide, PCO2, and y-axis is ventilation, right? Please note down that this can be given as an image-based MCQ. So what do you think? If your carbon dioxide is rising more and more in the arterial blood, what will happen to your ventilation? There will be more and more increase in ventilation. Note down. Question can be given like this, that one person is given inhalation of 2% followed by 3%, 4%, 5% carbon dioxide. Which of the following curve will be seen in this patient? This kind of question can also be given. So remember, whenever you are giving more and more carbon dioxide, there will be more and more increase in ventilation in a linear fashion. So note down, suppose the partial pressure of carbon dioxide in the arterial blood, what is the normal value? 40. Suppose the partial pressure of carbon dioxide is rising from 40 to 50 to 60 to 70 to 80. What will happen? More and more ventilation is going to increase. Ventilation is rising more and more, right? What part of ventilation you are talking about? Both depth is going to increase as well as the rate of ventilation. Both are going to be increased in this condition. But note down, this increase in ventilation will continue until and unless you are reaching at a carbon dioxide value of 80 millimeter of mercury. So up to this 80 millimeter of mercury, this increase in ventilation will continue whenever you increase the carbon dioxide. But whenever your partial pressure of carbon dioxide is beyond 80, what will happen? Further increase in ventilation is not possible. Rather, there can be decrease in ventilation also. So this can be seen in case of deep sea divers. Whenever the gas is inhaled at a high pressure, the carbon dioxide partial pressure also increases in your arterial blood. And if that partial pressure of carbon dioxide is beyond 80 millimeter of mercury, rather than stimulation to the peripheral chemoreceptor, it can causes depression of your peripheral chemoreceptor. So please remember, from 40 to 80, the increase in ventilation is linear. Means if you double the carbon dioxide percentage, your ventilation will also double, right? If you double the carbon dioxide percentage, your ventilation will also double. That is the meaning of this linear relationship. But this linear relationship is maintained up to 80 millimeter of mercury. Very, very important point. Now, if the carbon dioxide percentage or concentration is decreasing in my arterial blood, so it is decreasing from 80, decreasing. I can find out this point where carbon dioxide percentage or partial pressure is so low that your ventilation is zero. It is touching the x-axis. So what does that mean? That means the ventilation is zero. So ventilation is stopped for certain period, that is apnea. So this point where ventilation is temporarily stopped because of low carbon dioxide in the arterial blood, this point is known as apnea point. So question will be asked, at apnea point, what is the partial pressure of carbon dioxide in the arterial blood? Your answer will be 37 millimeter of mercury or lower, right? So whenever you reach at a partial pressure of carbon dioxide which is 37 or below that, then your ventilation will stop for certain period, and that is nothing but apnea, right? So that's why I told you that, look at partial pressure of carbon dioxide is arterial blood is 40. If the partial pressure of carbon dioxide is decreasing to 37, only 3 millimeter of mercury partial pressure has changed, but your respiration is completely stopping, right? Completely stopping. So that's why the carbon dioxide is very important point. And I told you already that carbon dioxide or hypercapnia, that is a stimulus for which chemoreceptor? Both. It is a stimulus for both central chemoreceptor as well as it is a stimulus for peripheral chemoreceptor. But how much stimulation is from central chemoreceptor? 80%, and for peripheral 20%. So you have already seen that this central chemoreceptor, they are directly stimulated by which ion? H plus ion. So if the question is like this, that for central chemoreceptor, okay, that for central chemoreceptor, which one is the direct or primary stimuli? So what will be the answer? Just now I told you that it is nothing but the H plus ion, but that H plus ion should be present at the level of CSF or interstitial fluid of brain. But if the question is most sensitive stimulus for this, okay, most sensitive stimulus for this central chemoreceptor, again your answer is carbon dioxide. Getting my point? So both peripheral chemoreceptor as well as the central chemoreceptor, the most sensitive stimulus is carbon dioxide, and that's why carbon dioxide is one of the important stimulus for peripheral as well as central chemoreceptor. So this graph you must have to remember. Now obvious question is, then what is the relationship between this ventilation and hypoxia? Right, so now we are going to discuss the relationship of ventilation and hypoxia. So again I will draw this graph. So x-axis will be obviously partial pressure of oxygen. So this is the partial pressure of oxygen, and y-axis is ventilation. Do you think you are going to get a curve, linear curve like that of the carbon dioxide? Answer is no. And that's why this has been asked in image-based question already. Right, now we look at the curve. The curve is very different from that of the carbon dioxide curve. The curve is very different from that of the carbon dioxide curve. Look at it. So when the partial pressure of oxygen is 160 millimeter of mercury, this is the partial pressure of oxygen, 160 millimeter of mercury, no hypoxia. Okay, suppose this is the resting ventilation, right? Obviously low, but again ventilation is there. Now suppose the partial pressure of oxygen has reduced to 100 millimeter of mercury. Obviously still there is no hypoxia. So although the partial pressure of oxygen has been reduced from 160 to 100, ventilation is not rising very much, very little rise. Even if the partial pressure of oxygen is decreasing up to 60 millimeter of mercury, I can find out that the ventilation is not rising to a very high level. Are you getting this curve? Although the hypoxia is there, 60 millimeter of mercury partial pressure of oxygen means from this 60 up to 100, okay, there is hypoxia, but that hypoxia is not stimulating your ventilation to a very high level. But after 60, suddenly there is increase in ventilation. That's why you can find out this curve, that if the hypoxia is more severe than 60 millimeter of mercury, okay, then ventilation is suddenly rising to a very high level. So question is, why there is not rise of huge amount of ventilation up to this 60 millimeter of mercury partial pressure of oxygen? So what is happening in this zone? Although there is hypoxia, but why that hypoxia is not proportionally stimulating your ventilation? So whenever there is hypoxia will stimulate the ventilation, right? But I am saying that ventilation is not rising proportionally up to 60 millimeter of mercury partial pressure. Because of this, that hypoxia will also causes ventilation. Ventilation increase means, okay, what will happen? There will be increase in carbon dioxide washout. Whenever there is increase in carbon dioxide washout, there will be production of alkalosis. Now you tell me, what is the effect of alkalosis on ventilation? It will try to inhibit the ventilation. So getting my point. So whenever there is more and more hypoxia is produced from this 100 millimeter of mercury up to this 60 millimeter of mercury partial pressure of oxygen, there is gradual more and more hypoxia that is trying to stimulate the ventilation. Other way, the alkalosis is trying to inhibit that ventilation also, right? Not only that, whenever there is more and more hypoxia will create more and more deoxyhemoglobin. You know, oxyhemoglobin is more acidic and deoxyhemoglobin is less acidic. Means I can understand that deoxyhemoglobin is like alkali. So it is like alkali. So if it is like alkali, what it will do to ventilation? It will also try to inhibit the ventilation. So during this zone from 100 to 60, hypoxia is trying to stimulate ventilation proportionally, but alkalosis and deoxyhemoglobin, they are trying to inhibit the ventilation within this zone, and because of this inhibition, the ventilation is not rising along with hypoxia. But whenever the hypoxia is below 60 millimeter of mercury, in this condition, hypoxic stimulation becomes so severe that the inhibitory effect of carbon dioxide washout and inhibitory effect of deoxyhemoglobin will not be able to prevent the increase in ventilation, and that's why I can find out that suddenly there is rise of ventilation to a very high level whenever hypoxia is below 60 millimeter of mercury partial pressure, right? So this way you have to remember the effect of hypoxia and effect of hypercapnia on your ventilation. Now I have to understand another curve where we have to combine two stimulus. Suppose hypercapnia along with hypoxia. What is going to happen, right? So I am drawing another curve here. Again, this curve is very important. So we have already seen that if x-axis is the partial pressure of carbon dioxide and if y-axis is ventilation, we have seen that the relationship between these two is linear relationship, but this relationship is only seen when oxygen partial pressure is normal. So suppose this curve is plotted when partial pressure of oxygen is 100, no hypoxia, only hypercapnia, right? Okay. Now if there is additional hypoxia in the body, so hypercapnia means carbon dioxide is rising in your body as well as hypoxia. What will happen? You will find out a curve which will looks like this, So here this curve, suppose the partial pressure of oxygen here is 60 millimeter of mercury. If there is more hypoxia, okay, suppose the hypoxia is more severe, then the curve will be more steeper. So I'm writing here PO2, suppose 40 millimeter of mercury. So what you are finding out, more and more hypoxia along with hypercapnia means the curve is shifting on the left hand side, means the curve is becoming more and more steeper. What do you mean by steeper curve? Means ventilation is rising. Previously also the ventilation was rising like this, but now additional hypoxia is rising the ventilation, which is more steeper rise than that of the hypercapnia alone, right? So that's why I can say like this, that hypoxia along with hypercapnia, right? What is the effect? More steeper curve, right? What is the meaning of that? Means more faster rise of ventilation, okay, more faster rise of ventilation. That means I can understand that hypoxia is increasing the sensitivity in response to carbon dioxide. Means carbon dioxide stimulus is there, but in presence of hypoxia, the receptor are becoming more sensitive, and that's why hypoxia along with hypercapnia is rising your ventilation, is stimulating your ventilation to a more severe level, to a faster level, right? So this is the meaning of this curve. So you can find out that all these curves are meeting at a single point. What is the name of that point? This point is nothing but the apnea point. What is the partial pressure of carbon dioxide at the apnea point? 37 millimeter of mercury. Now we have not considered another stimulus in this three curve. What is that stimulus? We have not considered what is the pH in this three curve. If there is acidosis, there will be additional stimulation. So suppose all this curve has been plotted with a pH of 7.4, right? Now, if there is additional acidosis, means hypercapnia, hypoxia along with acidosis, all the three stimuli are present in the body, then similar kind of curve will be seen. But look at, okay, so I have to draw this curve carefully so that this red color curve is parallel to the other red color curve that you are finding out here. So I am drawing in such way that these two red curve, they are parallel with one another, but it is shifted on the left hand side of the curve, means I can find out that the apnea point had shifted on the left hand side. What is the condition? Condition here, what is the partial pressure of oxygen? Partial pressure of oxygen is 100 here. But what is the pH? pH in this curve is acidic, that is the 7.3 is the pH. Similarly, if you add hypoxia, so I will take the same color, look at this same color curve should be parallel to the other curve which is present here. So what is the partial pressure of oxygen in this curve? This is 60. If I draw the third curve, okay, so this black color curve should be parallel to the other black color curve that you are finding out here. The partial pressure of oxygen is 40. So what you are getting? You are getting parallel shifting of the curve if there is additional acidosis, right? So acidosis means the curve will be parallelly shifted on the left hand side, means your apnea point will be shifted on left hand side. So in all these three curve, we have assumed that the pH is 7.3, that is the acidic pH. So bottom line is that if the acidosis is added along with this, then means hypercapnia and hypoxia, then the curve will shift parallelly on the left hand side, means your apnea point will be at a lower value. Okay, your respiration will stop at a lower value. It is not 37, it will be lower than that of the 37. So this curve has already been given in MCQ. So this exactly the same diagram is given in Guyton. So you can check the Guyton also, but not require. The same kind of diagram is given in Guyton. So this is all about the chemoreceptor and how they are functioning. Now after discussing this neural regulation, chemical regulation, so last that I told you already that we are going to discuss certain reflexes, and that is nothing but your Herring-Brewer reflexes. So first reflex that we are discussing, Herring-Brewer reflexes. So Herring-Brewer reflexes, this reflexes is also important because this is the first negative feedback reflexes discovered in physiology, okay, from human, right? First negative feedback reflexes which is discovered. So this Herring-Brewer reflexes, it has two components. One is Herring-Brewer inflation reflex, Herring-Brewer inflation reflex, right? And second one is Herring-Brewer deflation reflex, Herring-Brewer deflation reflex, right? So according to the Herring-Brewer inflation reflex, very simple, it is saying that inflation of your lung prevent further inflation. So inflation of your lungs prevent further inflation. Very simple. So if you do a deep breathing like this, right, what is happening? At the end of this deep breathing, I can understand that there is stoppage of breathing. So who is stopping that breathing? This is nothing but this Herring-Brewer reflex. So what is that? Whenever you are taking more and more deeper breathing, the stretch receptor of the lungs is activated, which I have already discussed in the neural regulation. I told you that there are stretch receptors which are activated when your tidal volume is greater than 2 liters. So please remember the receptor for this Herring-Brewer reflexes. Okay, so the receptor for this Herring-Brewer reflex, they are nothing but the stretch receptors of lungs. So where this stretch receptor of the lungs are located? Remember, they are located at the level of muscles. What kind of muscle? They are located at the level of smooth muscles of bronchi and bronchioles. Bronchi and at the level of bronchioles, right? And they are slowly adapting type of receptor, SAR, slow adapting type of receptor. For the time being, you remember slow adapting receptor. What is the meaning of slow adapting receptor? You will understand in central nervous system chapter. Okay. So when this receptor will be activated, remember that the activation of this receptor will be done whenever your tidal volume is greater than 1 liter, means double that of the normal. So whenever you are taking a deep inspiration, right? So there will be more and more inflation of the lung. Whenever your tidal volume during this inflation is beyond 1 liter, it will activate the vagus neuron, the myelinated fiber of vagus, and that vagus neuron will inhibit the inspiratory neuron, and that will prevent the further inflation or further inspiration. Getting my point? So this way, Hering-Brewer reflex is a negative or negative feedback reflex pathway. Inspiration, expansion of the lung is preventing further expansion. Inflation of the lung is preventing further inflation. Negative feedback reflex. So what I told, what is the afferent? The afferent is nothing but vagus neuron. But what fiber of vagus? Myelinated fiber of vagus neuron, right? So this is the information that you have to remember. Now what about Hering-Brewer deflation reflex? Very simple, just make it opposite. So strong deflation of the lung prevent further depletion. Otherwise there may be collapse of the lung. So if you go on compressing your lung more and more, one point will reach where this deflation, severe deflation, will prevent further deflation. So deflation of the lung prevent further deflation. Again, this is known as Hering-Brewer deflation reflex. Now question is, whenever you are doing deflation or whenever you are looking at this Hering-Brewer deflation reflex, what is the receptor? Because stretch receptor cannot be the receptor here. Okay, right? Because you are not stretching the lung, you are compressing the lung. So it is said in books that Hering-Brewer reflex, deflation reflex particularly, it is not due to the stretch receptor solely, mainly. It is mainly due to the proprioceptor. It is mainly due to the proprioceptor. Okay, proprioceptors which are present at the level of respiratory muscles and bones, right? So this is mainly mediated via proprioceptor, and there is some role of stretch receptor of the lung, but inflation reflex is the main reflex where the stretch receptor of the lungs are activated. For Hering-Brewer deflation reflex, the proprioceptors are mainly activated, right? So this is all about your Hering-Brewer reflex. But never forget the location of the receptor for Hering-Brewer reflex, right? Okay. Now, next is that Head's paradox reflex. Head's paradox reflex. This reflex was first time described by Henry Heads, name of the scientist who discovered this reflex, described this reflex for the first time in 1884, right? Henry Heads paradox reflex. So what is the reflex and why there is a paradox, right? But if you compare this reflex with Hering-Brewer, then you will understand the paradox. So it is saying that inflation of the lung stimulate further inflation. So what I am saying, if you inflate the lung, inflation of the lung is stimulating the further inflation. That is Head's paradox reflex. So what is the paradox? If this is happening in your body, what is going to happen? Lung will be expand more and more, and ultimately lung will rupture. So suppose I am taking a small inspiration and that inspiration is stimulating further. So what will happen? Lung will go on expanding, and a vicious cycle will be created until and unless there is damage of the lung. So obvious thing is that this Head's paradox reflex is not seen in adult lung, but there is only one condition where this Head's paradox reflex can be seen, and that is nothing but the first cry of newborn baby, right? First cry of the newborn baby. That means the reflex has occurs in your lung also, in my lung also, but it is not present nowadays, right? So whenever the newborn baby is taking first gasp of respiration, cry. So what is cry? Whenever you try to cry deeply, You have to take a deep gasping, and that deep gasping is nothing but an inspiratory effort. That inspiratory effort will stimulate more inspiration, lung will be fully expanded, and all the water which is there in the lung, that will be washed out, that will be squeezed out from the lung. So this way, the head's paradox reflex is very, very important during the first cry of the newborn baby, but it is not present in adult lung, right? And the last reflex that we are going to discuss in this regulation of respiration chapter is J receptor reflex, which is also known as C fiber receptor reflex. C fiber receptor reflex. So what is C fiber? You have seen in neuronal classification that there is three types of nerve fiber: A fiber, B fiber, C fiber. C fiber is the unmyelinated nerve fiber. Previously I told you that if you look at this Herring-Brewer reflex, they are the slowly adapting receptor, and what is the afferent? Afferent is the vagus neuron. What kind of vagus neuron? Myelinated fiber of vagus neuron. If you look at the J receptor, you know that the afferent from this J receptor is also vagus, but the unmyelinated fiber of vagus, and this unmyelinated fiber is the C fiber. That's why the J receptor reflex is also known as C fiber reflex. Getting my point? So this J stands for juxta capillary receptor. So what is the location of this receptor? So first important question, what is the location of this receptor? Okay, if this is an alveolus, you know that just outside this alveolus there will be capillaries. So suppose this is the pulmonary capillaries. J receptor means they will be located close to the pulmonary capillary, but close to the pulmonary capillary means it is not on the capillary wall, outside the capillary wall, close to the capillary. Mostly they are present on alveolar wall, or they may be present also at the level of lung interstitial tissues. Getting my point? So location of this J receptor is alveolar wall or lung interstitial tissue close to pulmonary capillary, right? So this important information you have to remember about J receptor or juxta capillary receptor, juxta pulmonary receptor, right? So this is J receptor. Now remember, J receptor are never activated by physiological stimulus. J receptor activation means all the stimuli are pathological stimuli. So it has no role for your resting breathing or eupneic breathing, normal breathing. The J receptor has no role. So when this J receptor is stimulated, okay, stimulation of this J receptor, right? Okay, so stimulation of this J receptor, number one, if there is engorgement of these pulmonary capillaries, okay, so suppose there is high pressure engorgement of pulmonary capillary, okay, then this J receptor can be activated. Or there may be accumulation of water at the level of lung interstitial tissue, so that is edema, okay, or ARDS, then also this receptor will be activated. Or suppose there is pneumonia, okay, or if there is embolism, then also this receptor will be activated. Or if there is hyperinflation of the lung, very huge expansion of the lung, hyperinflation of the lung, then also this receptor can be activated. And number 6 is certain kind of chemical like capsaicin can also activate this receptor. These receptors are similar to the coronary chemoreceptor reflex. The behavior is similar to that, okay? And the functioning of this receptor was first time discovered by one Indian scientist, and he is the famous A.S. Paintal, right? So please try to remember this name, A.S. Paintal. In 1954, he first time discovered this receptor, Dr. Pentel was alumna from KGMC Lucknow, right? So he for the first time described this J receptor, and this J receptor is activated by multiple pathological stimuli that we have discussed here. Whenever this J receptor is activated, what is the response, means what is going to happen in case of patient. So what is the responses when this receptor is activated? Responses are very, very dramatic. What dramatic? So there will be hyperventilation, okay, there will be hyperventilation, rapid shallow breathing will be there, rapid shallow breathing, followed by there will be apnea, there will be bronchoconstriction, there will be bronchoconstriction, right? There will be increased mucus secretion, okay? So all this thing ultimately will leads to dyspnea, right? You can easily find out. So rapid shallow breathing followed by apnea followed by bronchoconstriction, okay? Increased mucus secretion, along with there will be decrease in blood pressure, decrease in heart rate, okay? So hypotension, bradycardia is also going to happen along with this hyperventilation and apnea and bronchoconstriction. So these are the symptoms which are produced when the J receptor is activated. That's why it is seen, even if there is a small emboli at the level of lung, suppose that emboli is not going to cause severe hypoxia of the blood, but still that embolism can activate the J receptor, and whenever your J receptor is activated, severe dyspneic like symptoms can happen due to this J receptor response. So this way you have to remember that J receptor is activated in various pathological condition, and whenever this receptor is activated, dramatic symptoms like this hyperventilation, bradycardia, apnea can happen. So this is all about your control of breathing or control of your respiration.